NEB TV. What's trending in science?
Welcome to NEB TV. Today I'm joined by Alicia Bielik, who is a group leader on our production team. Hi Alicia.
Hi Dina.
And what are we talking about today?
Today I'd like to introduce you to the importance of glycobiology. And then you will hear from two experts in the field, Pauline Redd and Gordon Lauk, on the importance of clinical and diagnostic glycoscience, as well as pharmaceutical glycoscience. Stephen Shi and I will then describe the breadth of NEB's glycobiology product line and our commitment to the highest quality standards.
Great. Are you ready?
I'm ready.
Let's get started.
Glycobiology is the study of the structure, function, and biology of carbohydrates, which are also called glycans and are present in every living organism. It's a rapidly growing field in biology with relevance to biomedicine, biotechnology, and basic research. Proteomics has expanded the knowledge of protein expression, modification, interaction, and function. In eukaryotic cells, the majority of proteins are post-translationally modified. One of the most common modifications, which is essential for cell viability, is the attachment of glycans, which is also known as glycosylation. These glycans are diverse, complex molecules whose function is determined by their unique structure. Effective enzymatic tools and analytical techniques are required to properly elucidate glycan structure, which then can be correlated to a specific function. Glycosylation defines the adhesive properties of proteins in cells, and in fact, the immune system largely functions via glycan-protein interactions. This highlights the importance of glycans in physiology, pathogen recognition, cancer, and autoimmune diseases. Considering the central role that glycans play in immune interactions, glycoproteins and carbohydrates are becoming the next generation of therapeutics, vaccines, and diagnostics.
Most of the new biological drugs are glycosylated proteins. The glycans have a number of really important functions in the role of those proteins. So, for example, in erythropoietin, which is probably the biggest blockbuster drug at the moment, needs the correct sugars attached to the proteins in order to maintain a useful half-life for the protein. If you have the wrong sugars, it will be cleared in 3 minutes. If you have the right ones, it will be in the patient at least 3 hours, in which case it has time to act in its biological role. Monoclonal antibodies also have sugars which modulate the effector function of the proteins. So they're generally targeted to epitopes on cancer cells, for example. When they engage with those epitopes, it's really important to control the next stage of the process, which is for the Fc region of the molecule to bind to some other receptor and then to activate a biological event such as antigen-dependent cellular cytotoxicity. We also know that glycans can be pro or anti-inflammatory molecules, so all these drugs are designed to act with the patient's immune system. And if, for example, the IgG molecules have sugars ending in sialic acids, then these are basically anti-inflammatory molecules. So it's very important to have an understanding of these different roles and functions of the sugars before you design the kind of antibody glycosylation that you need for your particular purpose.
Glycans change both with healthy aging and with disease. In the last few years, in Genus, we have analyzed over 50,000 individual glycomes, and in the majority of studies, we found significant changes. In many diseases that we studied, including diabetes, cancer, and IBD, changes in the composition of either total plasma glycome or the IgG glycome contained more information about the disease than all other clinical parameters combined. Therefore, glycan profiling is definitely a very promising biomarker.
It's very important to profile and to have a detailed analysis of the sugars attached to your drug. For example, you need to be certain that there are no antigenic epitopes, because sometimes if you make a drug in a non-human cell line, you may end up with an epitope on the sugar which will react with the immune system and cause a very severe response. And since this has happened a number of times, the regulators really press home the importance of checking for this. It's also important to monitor the glycosylation because it is the end point of any production of a glycoprotein, and if it's consistent, it's telling you that the bioprocess itself is consistent.
Clinical glycomics is clearly heading towards a new revolution. Analytical methods are improving a lot, and nowadays they enable routine high-throughput analysis of both total plasma glycome and some individual glycoproteins. This was very clearly demonstrated in a recent large collaborative study that was performed by GENOS, LUMC, NIBRT, and MPI. The availability of technology and the first large clinical studies that were published in the last few years are also raising interest among our clinical colleagues, who are showing higher and higher interest to include glycans in their studies. The field of personalized medicine is surely one big area where glycomics is expanding. Diagnosing and monitoring diseases is currently in the focus of research, but even more promising seems to be the development of companion diagnostics that will help predict response to different therapeutics. Since glycome is both highly heritable and highly versatile at the same time, it seems to be an ideal tool for stable patient stratification, which is exactly what is needed for precision medicine.
Our focus now is on further miniaturization. We would like to see it transferred onto a chip, for example, so that it could easily fit in at line in a manufacturing process. We'd also like to see the analytics being quicker, faster than the current LC-MS system. And I think those are two major targets. The high throughput robot, which we use to do multiple releases simultaneously, is also going to be really important because people are now moving towards establishing a human glycome project, which will be looking at the glycosylation of serum or individual molecules from many thousands of people.
As you have heard from leading experts, glycosylation is increasing in its importance to regulated industries like pharma and clinical diagnostics. Many glycosidase manufacturers are still using practices designed to support the research market. Recently, we tested analytical glycosidases from several commercial providers for various quality attributes. We find a range of issues that highlight the need for revised manufacturing practices for glycosidases used in pharma and clinical applications. For example, 1-alpha galactosidase from one supplier produced from coffee bean turned out to have substantial beta galactosidase activity. In another case, 2 of the galactosidases from different suppliers we examined were found to contain sialidase activity. The presence of unwanted enzyme activities can obviously taint experimental results, especially when glycosidases are used alone or in combinations to elucidate or sequence elaborate glycan structures.
The quality of NEB's products is both an NEB strength and a differentiator from other providers. It's our fundamental promise to both the scientific community and every NEB customer. We have a comprehensive quality management system that ensures every step in the manufacturing process is held to the highest quality standards in the business. One indication is our certification to ISO 9001 quality management system, as well as ISO 13485, which is a requirement for the medical device industry. NEB is also building a GMP facility, which, when complete, will elevate our quality standards for certain products even higher. We have created a set of stringent quality control standards for every enzyme that we produce, including a protein purity standard of greater than 95% pure, and a sensitive assay that includes a panel of 18 substrates for the detection of contaminating exo or endoglycosidase activities. Each enzyme is also tested for the absence of protease activity. In this way, we are able to manufacture enzymes with unmatched quality standards and exceptional lot-to-lot consistency.
So Alicia, thanks so much for joining us today.
Thanks for having me.
And as always, if you have any suggestions for future episodes, please let us know.